Today we are going to, I believe, finish our discussion about genetics. And in our discussion of genetics, we're going to answer perception, because that's part of what I use as education. That's where we're headed. So that's kind of the overview for today. We're going to talk about how DNA becomes a protein, how the DNA can be replicated so that the cell can divide, and then we'll talk about how that division of cells gets out of control. So that's our end goal for the day. Okay? So as we mentioned last time, it's all about genetics. I changed the format of the slides. Did you notice? I'm not sure I like it yet. We'll decide later. But anyway, we are all about genetics, and the quiz is there now available for you. It wasn't there Monday, but I tweaked it, added some questions that I didn't like, added a couple I didn't like, so it's all ready to go. There is also a set of videos that are right above the quiz in the modules. Remember modules? That's the way I organize my life. That's the way I expect you to be looking at things. So if you look at the quiz, you'll see a set of videos right above it. And I'm encouraging you to look at those, watch those videos before you attempt the quiz. There's a lot of good information in there. Some of you like to watch videos, TikTok. So why not, right? They're not TikTok videos, but still they work. And then there's a couple of Connect Labs that I have to brush up and make sure those work. And then the class will go lab will go to genetics. Okay? So what ought I know before I'm done today? I should know the overall mechanism of the production of a protein, where we left off last time. So we're going to talk about the production of a protein starting from DNA to the final product. You need to know the molecules, the functions, the enzymes, and the locations. Got graphs, pictures, etc. You should know the overall mechanism for DNA replication, not the nitty-gritty details, just the overall. And how that then leads into the cell cycle and what occurs in the cell in each stage, and do all cells complete it, and what cell types do not. Then talk a little bit about growth factors, something called proto-oncogenes, something called tumor suppression genes, mutations, tumors, as I mentioned, what causes mutations, something called angiogenesis, cancer cells, how they differ, and then what causes the tumor to present the problem. So that's our goals for the day, and that's where we ought to be when we're done. So when we were together last time, I set out to say these are the objectives for the overall unit. How do molecules and processes transfer characteristics from cell to cell? We'll get into that more today. How do they allow for the repeated expression? Again, we're going to dig into that more. And then how do changes occur? As I said, I'm going to use mutations specifically leading to cancer, but the overall process of mutations is what we believe to be the driving force for evolution within a species. So that's where we're headed. All right, so last time we outlined this molecule, the DNA molecule. And as I said, it gets packed into the cell. It gets associated with proteins to be able to condense it down to a small enough structure that we call a chromosome to fit into the nucleus of the cell. Remember, that led to some of the history that was taking place. DNA, or was it the protein that was the actual molecule of inheritance? And as we mentioned, how we know it is DNA. The protein's there just for packaging. So what is this DNA? It's a double-stranded molecule. So there are two backbones, what we call the backbones. The backbone is sugar and phosphates. The sugar is deoxyribose. And each sugar is linked to the next sugar through a phosphate group. And projecting off of the sugar is the nitrogenous containing base, nitrogen containing base. 4 different flavors: adenine, thymine, guanine, cytosine. And we know that adenine and thymine pair together. If there's an adenine on one strand, there is thymine on the other strand. Same with guanine and cytosine. Because of the structure of those adenines, thymines, guanines, cytosines, as you notice, they are both the same distance apart. It's a straight line. Okay? It doesn't bulge, shrink, bulge, anything weird like that. For every T, there's an A. For every G, there's a C, directly opposite. Okay? And those 2 strands are held together by hydrogen bonds. The weakest of our bonds. We're going to take advantage of that in a second. All right, so that's where we left off. And we said we are going to get from this gene that's found within the DNA, and we're going to make a protein. And it's not just a random thing. A gene is a specific, discrete unit of DNA on a chromosome. And it has a specific start, and it has a specific stop. All of the nucleotides in between are the information that will be used to make the protein. Start is always a UG on the messenger RNA. That leads to a methionine. Very first amino acid in every protein. Stops, there's 3 of those. All the other groups of 3 codons code for or have the information for all the other amino acids, for the other 19. Okay, 20 minus methionine. So that's where we're headed. And as I said, there's going to be 2 processes. This was from last time. With 3 types of molecules. So we're going to go from DNA to RNA to protein. And as I mentioned, And as I mentioned, DNA and RNA, both nucleic acid language. Both are nucleotides. DNA, deoxyribose, RNA, ribonucleic. They are the same kind of language. You know, it's just that you write your T's differently than I do, or something like that, right? That you are copying faithfully. So that is the process we call transcription. Then we're going to take the nucleic acid language of RNA and we're going to make a protein. Now we are going from nucleic acid language to amino acid language, and that requires transcription. Change the language. So transcription is DNA to RNA. And the most important enzyme that is required for transcription is called RNA polymerase. Now, stop and think, what does that enzyme tell us it's doing? Because the name of the enzyme tells us what it is going to do. So, a polymerase, what does that sound like it's going to do? Do what? I can't hear. Go ahead. Not erase. Although I get why you said that. You're right with the polymer. You're absolutely right with the polymer. What's it going to do to the polymer? It's going to make it. All right? Make the polymer. What is it going to make? An RNA polymer. Right? So it's making. I see why you said erase. I mean, it's right there in the word, right? So it's going to make a polymer of RNA. That's what it's intended to do. So it's going to copy the DNA to RNA. Remember, we're staying in the transcription language. We're just transcribing. Taking nucleic acid, we're making a nucleic acid. The one exception, other than the sugar, deoxyribose in DNA, ribose in RNA, the one other exception is that wherever there's a T, instead in RNA it's a U, uracil. That's it. Okay? True statement. Kind of a test question, right? True statement. What molecule has uracil? DNA or RNA? You will always answer RNA. What molecule has thymine? DNA or RNA? You will always answer DNA. Well, here we go. Okay, what molecule has guanine? DNA or RNA or both? There we go. Okay, catching on. Now, when we are done with this process, we create, or what is created, rather, is called an mRNA, messenger RNA. So the DNA has the information. It's been copied into this new form called mRNA, messenger. Now we have the information. Just a small little section of DNA, that is the gene. So there's the gene, there's the RNA, there's the messenger RNA, and now we're going to get ready to do translation. Now where's the DNA found? It's in the cell, but where in the cell? The nucleus. Where is a protein produced? Inside the nucleus or in the cytoplasm? Cytoplasm. So guess what the mRNA has to do? Get out of the nucleus. Okay. So it's able to leave. If you recall last time I had that little table and had some of those compares and contrasts. That was one of them. Okay. All right. Now this is called pre-mRNA because what's happened to our genome, that is the collection of all of our DNA, over time, there are little pieces in our genes that are not to be used for protein production. They are called introns. Now, intron means intervening. I don't know how they got from intervening to intron, don't ask, I don't know. But they are not part of the actual translated messenger RNA, final product. They're not part of it. So they get clipped out. And you end up with that. What is the reading, the information that we need is called exons. Expressed is where that comes from. So that's the final product. Okay? That's what we're going to use to do protein production. So there's a whole lot of nucleotides in your DNA that we're not quite sure what they're doing. We have various reasons why they're there, but nobody's settled on what the purpose of the intron is. So, bigger description, there's the DNA. There's the transcription, pre-mRNA. There's the mRNAs. Okay? There's going to be the protein. That's what's happening. So, translation is taking the RNA, nucleic acid language, and we're going to translate it into a new language. We're taking your English and making it Spanish. You are taking your German and making it English. Whatever you are doing. You are going to translate it. Now, the translation is RNA to amino acid. As I keep emphasizing, it makes sense for most of my students over the years to say the language changes. That's how I kind of keep it straight. Nucleic acid, nucleotides to amino acids. 3 nucleotides at a time. AUG means methionine. 3 nucleotides, AUG, in order, means the amino acid methionine. Just for an example. Okay? That's what we are trying to get across. Now, There are other types of RNA, not just messenger RNA. There's the message. But how are we going to translate it? Well, we're going to use something called transfer RNA, tRNA. What that's going to do is bring the amino acid, correct amino acid, to the added. It's going to bring the correct amino acid to grow the chain. I can't get past that one point. To grow the chain of amino acids. It's going to bring the correct amino acid in the primary sequence. Remember our structures of protein. We start with just naming the amino acids. This is bringing the correct amino acid to build the chain. It will have on it what's called the anticodon. Hang on, it's better to look at a picture. You can write it down, but hang on, I'll explain with a picture. Now, there's another one. So there's the message. There's going to be a transfer RNA to transfer the amino acids. And we have to have some place for all of this protein production to happen. Remember, you were asked to know some of the organelles. One of the organelles that I asked you to know is a ribosome. The ribosome becomes the platform for protein production. It's where the action happens. It's the stage, whatever gets you there. It's the field of play. Court, I don't care, whatever. Now, the ribosome is made of our third RNA. Ribosomal RNA. Plus some enzymes. We call that the ribosome. So, in the process of translation, how many different RNAs are present? 1, 2, or 3? Process of translation, how many different RNAs are present? 1, 2, or 3? All 3. We'll see in a second. All 3 are present in the process of translation. Because we have the messenger RNA, that's the message from the DNA. We have the transfer, bringing the amino acids. And we have our RNA, ribosomal, we're going to take out. All 3 are present. This is transfer RNA, wacky, weird molecule. Okay? Looks weird. This is where the amino acid binds. So there is a separate transfer RNA for every amino acid. There's a pool sitting in the cell. There's the anticodon. Now if that's UUA, uracil, adenine, what is on the messenger RNA? Not one. Adenine, uracil. Thymine, because what? It's RNA. So it looks like this. Whoa! Look at that. A whole big process. All spelled out for you. There's DNA. There's the messenger RNA being made. It's leaving the nucleus. Ribosomes attached to it. Transfer RNA comes in. There's the codon and the messenger RNA, CAU. What pairs with C? G. What pairs with A? U. What pairs with U? A. There's the amino acid that should be there. GCA, CGU, there's the growing protein. That one is going to attach right there. That is how we are going to get the primary sequence. Naming the amino acids of a protein in order. This is how it happens. how it happens. Now, I don't think it's too far of a stretch for you to look at this and say, well, what if this were messed up? What if it wasn't CAU but was GAU? Would that be the same amino acid there? Would the protein potentially fold the right way? Potentially it would not. And if it didn't fold the right way, would it potentially do the right action? Nope. Sometimes it may actually do it better. Sometimes it may not work at all. Mutations. That's what a mutation becomes. It's something in the DNA that gets altered. Leads to an incorrect sequence of amino acids in the protein. The protein doesn't work the normal way, or the usual way would be a better word. Sometimes it works better. That's good. That's good. So, I would ask that you know these steps. Now, I'm not worried about the ribosome protein part over here. I'm more worried about this, to this, to this. Okay. You know those steps, you're on track. I don't want you to take pictures. It's on PowerPoint. It's online. That's okay. What's that?
I don't have to get online.
Oh. Now, if you like it a little bit more straightforward without all the other stuff, we have this diagram. Try to help you out. Okay. So there's the DNA. There is our template. The information that we're going to use to make a protein. There's the messenger RNA. TAC, AUG. AUG means methionine. Very first one. All the way down to other. Stop. What? Yeah, that's why it stops. There's no transfer RNA for that. If there's no transfer RNA, then there's no amino acid to put there. Stop. We're done. So if you like it this way, schematically, straight down the screen, hey, that's perfect too. Transcription, translation. Okay? What I'm asking you to know. The process has all three amino acids, or all three RNAs present, transcription or translation. All right, questions? Because we know this, we can now begin to understand protein structure, how proteins get dismayed into genetic issues. Okay, that's what the bottom line is. All right, I'm going to shift gears a little bit. A little detour for a while. We'll come back to DNA in a second. It's called the cell cycle. I'm not asking that you know everything that's going on in the cell cycle. Got a whole class of it. Take it. Cell biology. All I ask that you know there are 4 stages. One called gap. One, growth. Followed by DNA replication. We're going to come back and spend some time there. Then gap 2, or grow. Mitosis, ending with cell division. 1 cell becomes 2. Okay? So we're going to talk about this DNA replication bit right here, synthesis. That's where we're going to focus for the next few minutes. So 1 cell becomes 2. That's the big idea here. 1 cell becomes 2. 1 cell becomes 2. We need this to happen for growth of an organism. We need to add cells. And I mean, yes, the oocyte, your egg, is the biggest cell in the human body in terms of cytoplasm content. You're a whole lot bigger than that now. You are trillions of cells. So how in the world did that happen? We had to grow it. We had to add cells. We're going to add cells by cell division, this process called cytokinesis. Cell division. That's all that means. Now, every one of these new cells, whichever diagram I look at, that next statement has to be true. Each new cell must have the same genetic code as the original. Remember, I said there were 3 objectives we were going to try to accomplish. I said one of those was that traits, characteristics have to be passed on from cell to cell and organism to organism. Right here is how characteristics get passed on from one cell to the next. Your skin cells remain skin cells. They don't suddenly become something else. At least they should. But every one of your cells in your body has the exact same DNA because of this process. Why is my skin different than my liver? Because different enzymes, different proteins rather, are expressed. Different products. So, the DNA has to replicate for what we call the daughter cells. So you have a parent cell and 2 daughters. This is accomplished by what is called semi-conservative replication. Now, there are other ideas, but this is what we know to be true now. What does that mean? It means one parent is going to add or have paired with it a new strand. So we have here a parent and a daughter, parent and a daughter, or new strands. This is the original. We're going to add that new strand to it. We do that twice. So we have complementary pairs, A to T, G to C. That is how it's accomplished. That is how it's accomplished. Because of that faithful pairing of A to T, G to C. There are enzymes involved. The first one is called DNA helicase. Remember, DNA is a helix. Like helios outside. It's twisted. Well, you're not going to get very far if you can't untwist the twist. So helicase unwinds things, straightens it out. We're going to need RNA polymerase. You go, wait a minute, we're making DNA. And I'll show you why. And yes, we're going to need DNA polymerase. Why? Because what does this say? Polymer. We need to make a polymer. Polymer of what? DNA. Isn't that what we're doing here? We're making new strands of DNA. And we need something called ligase. Ligase means to knit, to seal, to heal, if you will. So there must be some sort of little breaks that we have to fix. Okay? And then we'll talk towards the end about mutations, why and how they can happen. That's our overview here of DNA replication. Ready? Here's a big overview. Simplified overview. So there's the DNA. That's what we're going to make a copy of. Now, DNA is this weird molecule. You can't just add DNA directly to it. So RNA is what's used to start it, called an RNA primer. That's why RNA polymerase is involved. Okay. If you like this sort of thing, I'll step off stage for a second. We're not sure if DNA or RNA was the original molecule of inheritance. Some people think it was RNA. And this is kind of indicated. Back over here for replication. So that's how it's going to start. Then we're going to get DNA polymerase to continue replicating. That's the primary enzyme that's going to add all of the new DNA nucleotides. Then you have to come back and you have to get rid of those RNAs. That's a little break right there that the ligase needs to fix. Another picture. Woo! That's all the enzymes. So here's our original DNA, parental DNA. Here's the helicase, unwinding. Here's the DNA polymerase, trucking along. Here's the ligase, fixing it where there's breaks. Up above, DNA polymerase. I'm not worried about which one's which, 3', 5' directions. I don't care. You don't care. I don't care. We're just going to skip that. Okay. All right. So, enzymes, helicase, DNA polymerase, ligase, and RNA polymerase. Now, showing this incoming set of nucleotides, and they're supposed to pair up. So on the original parent, wherever there's an A, what gets put into the other strand? Wherever there's an A, what gets put into the new strand? T. A to T. Wherever there's a C, it gets a G. It will do, DNA polymerase, will do this about 99.99% of the time correctly. 99.99. That 100%. Nope. But we have another enzyme. You don't need to know the name of it. It's kind of a proofreader, and it goes along and it says, "Ooh, that was wrong. Instead of the T with the A G got put in. Clip it out, replace it with the correct one, and it'll correct. About most of those, but we still have like 1/1000 of an error rate. But guess what? We've been around for, a while. So that little error rate must not have been that big a deal to us as a species. We continue just fine. Okay? But that little error rate can lead to a new protein, a protein that was not made the correct way. That's why we worry about replication a little bit. Now, as I said, the cell cycle starts with this G1. It's called gap, but it's really where growth occurs. Then the S phase, that's where DNA replication occurs. Then G2, which is technically gap, but what happens more growth. And then we get mitosis. Now, as the cell grows, it has to reach a certain size. Certain conditions have to be met before it'll replicate the DNA. And then the cell grows until it reaches a certain size, certain conditions are met, and then it will go through mitosis, where it's going to divide. And that way it is controlled. Certain conditions have to be met. It has to grow to the right size. Conditions for division have to be right, and then it'll undergo division. If something's not right, it shouldn't divide. That's the bottom line. Now, some cells don't go through cell division. Now, sometimes the cell doesn't go through cell division, which is called the G0, because the conditions aren't right. It just kind of hangs there and waits until conditions change and it's correct. But you have two cell types in your body that do not undergo cell division. They are basically the same ones that you were born with. Whoa. Neurons, cells of the nervous system, the neurons, do not undergo cell division. It's kind of a bummer because if you cut a nerve, it doesn't regenerate. Okay? I still don't have much feeling in the end of my finger where I sliced it off and took off some of the neuron. The big idea is spinal cord injuries do not repair. The cells cannot replace themselves. They can't repair. The other one, cardiac muscle cannot repair itself under most conditions, under most situations. Heart attack, tissue dies, tissue's dead. Not coming back. Heart's going to be a little less functional, a little weaker. Now, you really want to go into some research, you fix those two problems, my goodness, you're going to win Nobel Prizes and the accolades of everybody. Because we can't figure those out. We haven't got there yet. All right. So these conditions are called checkpoints, just to show you that's what has to happen. And notice, final preparation. See, it has to grow. Things have to be right. All right. So mitosis. Why do we need mitosis? I've already stated this. You are a one-celled organism. You are not anymore. You have to go through cell division. Add cells, add cells. Every cell has to have the same 46 chromosomes. That's why we need DNA replication. And because of that, tissues have to grow. I wish my tissues didn't keep growing, but they keep growing sometimes, and then they shrink, and then they grow. But sometimes they look really good. Oh, come on. You guys can smile a little bit. And then, you're not there yet, but your cells start to die. You know, you have cells dying all the time. Every 7 days to 10 days or so, you get a whole new layer of outer skin. Every 3 to 5 days, you get a whole new lining in your stomach. Because they need replacement. Then over time, stop replacing these cells as quickly. If I haven't said it, you're at the best point in your life you will ever be. After this, it's all downhill. Isn't that a great story? Somebody else in the room is going to sit there and go, yeah, well, you guys keep me young, or at least make me feel young. All right, but then there's injured tissues, right? You know, wouldn't it be great not to walk around with this open laceration? So we repair things. So you don't need to know TMAT. I'm not going to ask you to what is the order: prophase, metaphase, anaphase, telophase, and is there a pre-prophase and all this and blue mud bug? No, you don't need to know. I just like to show you pictures. That, to me, I hang that on my wall. I think that's kind of pretty art. A little abstract, but my wife doesn't go for it. She likes the more classic stuff. But what's going on is there's the lining up of the chromosomes, then they're pulled apart in telophase, and now we get the new two cells. It's just showing one cell becoming two. Now, this cell cycle is regulated. Remember, I keep saying, you've got to grow to a certain size, conditions have to be right, and then you move on, the cell moves on to the next stage. Well, how does that stay controlled? How do we get a control mechanism for this? One is called cell contact inhibition.
When your cells divide, at least in petri dishes, which we can do upstairs, they'll grow across plate, petri dish, when they reach the edge. They won't keep dividing. So the cells, when they're in contact with each other, kind of have this signaling back and forth. Okay, don't divide. It's not your turn or it's not your time. We're connected to each other. We don't need to go through this cell division thing. No damage, nobody's aged, nobody's worn out. Don't divide. No, they're not talking, but you get the idea. But if I put cancer cells in that same petri dish, oh yeah, they'll grow across the bottom of the dish, they'll grow up the sides, they'll start folding over, they'll make multiple layers, they just keep on growing. Because they've lost contact inhibition. Not thinking. Secondly, there are growth factors. Hey, that tells me what it's doing, right? G1, S, G2, what's going on? The cell is growing. Got a signal for it. Something to tell it to grow. These are all a set of proteins. There's a whole slew of them. More being discovered practically every month. Then there are things within your DNA that have special names. One is called proto-oncogenes. Now, I rule the world, which I don't, but I did. I changed the name. Here's why. Proto means before. What they did is they discovered something called the oncogene first. So we know what gene is, that's a segment of DNA. What does the prefix onco mean? Anybody know? Cancer. Because if you are visiting an oncologist, it is a specialist in cancer. So onco means cancer. So they found genes that were known to be causing or involved in cancer. And then somebody bright said, what did it do before it became mutated? Oh. So then we slapped the phrase proto in front of it. These make some of those growth factors and some of the receptors. And this is a great analogy that the oncogene, the proto-oncogene, and the oncogene are the gas pedals. Grow, 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 right? Keep growing, you're doing fine. But if the gas pedal gets stuck, cell division, no control. So the oncogene is the mutated version, and it's telling the cell to keep growing and dividing and growing and dividing. Now, we named this one pretty well. We said it's a tumor suppressor. That's its normal role. So, as our little diagram up there is like, it's like the brake. It's like the brake. So I live in the country, driving burnout marks all over the roads and certain places. Right? So you've never burned out a car. Some of you are smiling, so evidently you know how to do it. But you got to hit the gas real hard and hold on to the brake. And the tires are spinning like crazy and there's rubber on the brake. Well, what if the brake suddenly is inactive? Off you go. Okay? That's what's going on with these tumor suppressor genes. They are causing the cell not to divide until conditions are right. Take away the brake, and they just keep on dividing. So we have gas to start them growing, and we have a brake to keep them from growing too much. When those are disrupted, we get uncontrolled growth. So that's what a cancer is. Now, cancer. One of the astrological signs, right? There's 12 of them. I'm a Capricorn, whatever that means. just means when my birthday is. It's in that unit. That's about all it means. But some of you are born under the sign of Cancer. What is the sign of Cancer? Anybody know? Everybody reads the astrological charts every day to find out what's going to happen to them that day? Good. Yeah, not that. I want there's animals associated with them. Crab. It's a crab. So guess what? The early anatomists, people looking at stuff, looked at this tumor. They said, wow, it's got little arms that project out like this into the normal tissue. It's like the crabs that we see on the beach with all their little arms. So that's how it became cancer. Now, technically, a better term than cancer is neurocancer. Literally translated as new tissue. This is a tissue that was not there, it is now new. Or we could call it a tumor. You want to go all Arnold Schwarzenegger. But tumors are these new growths of tissue. There are two kinds. One you want, one you don't. No, you don't want either one. But one's better than the other. If you have to have one, you want one that not the other. And you don't want them, but if you have to, right? So the first is benign. Benign simply means that it is not spreading. It's growing, and it's staying there. And when we look at it, we find that there's kind of a membrane, a barrier around it. So if you're the surgeon, you open up the tissue, see the margin, see the border, you go cut, take it out, you're almost guaranteed to get a gulp. Because it's right there. Malignant, malignant means it can spread. Doesn't mean it will, doesn't mean it did, it can spread. That border, not necessarily present. And what's dangerous about it is metastasis. It can spread other tissues. How does it do that? I'll show you in a second. So here's normal. Starts growing a little rapidly, starts becoming different, and then it becomes disease. So if we look at tissue that is dysplasic, we know that genetically it does not appear the same. It does not look the same under the microscope. It is changing appearance. Now, what causes them? First and foremost, carcinogens. Carcinogens will cause cancer. Or in other words, a mutagen. Causes a mutation in the DNA. Chemicals that are found in anything that you smoke. I don't care what it is, you smoke it, there's something in there that will cause a problem. Don't buy the story that pot does not cause it will. Just haven't done it long enough. Now it's legal in too many states. It's going to start popping up. Guaranteed. Or it's something that you are exposed to. With the chemicals, not allowed to exposure to. Radiation. Radiation is high energy. High energy causes DNA to mutate. Virus. Yeah, viruses. There are certain viruses that can cause cancer. Now, some of the oncogenes, they go by fancy names like CIS, RAS, and B2. And then there's mutated tumor suppressors, RB, P53. This P53 down here, now I'm looking at my watch and I know I've got at least 1 minute. P53 is found in almost all colon cancers. So what does it do? It will displace normal tissue. Right? It's going in there and it's altering normal. It increases in cell number, which causes the tissue to decrease in what it can do. The metastatic tissue is not able to do the function. So here's the normal here's the tumor, and it gets into the bloodstream or into your lymph system, and it gets out into a new tissue, spread. This is a diagram showing a tumor of the breast tissue and the location of the usual sites where it goes. We know that the breast cancer is metastatic. They start looking in the lungs, the bones, the liver, and the brain. My mother-in-law had breast cancer. She had colon cancer. And she had a brain tumor. I'm not sure which one was which. That tissue does not do it. The brain's not going to function like breast tissue. It's going to cause blockage. It's going to cause increased metabolic rates. Tumors no bigger than my thumb kill people. The metabolic rates are all their energy being used by that tumor. Okay. Now, angiogenesis is when the tumor causes new blood vessels to grow to it. These suckers can cause blood vessels to grow to it. Now, they have a ready supply of nutrients. That's a retinoblastoma. That's RB. Tumor of the retina. And that, of course, that was powerful. All right. Thank you for your patience.